Welcome to this lecture, and this is for my course, Microorganisms and Their Human Hosts. This lecture is about protozoa and fungi. Now, we usually kind of default to talk about the bacteria and viruses as far as microbiology goes. However, I do want to introduce you to some eukaryotic organisms because of the 20 most frequent microbial causes of death worldwide, 6 are from eukaryotes. So there are 3 groups of eukaryotes that we're going to discuss, 2 in this lecture, and these 2 are going to be the protozoa and fungi. Now this is an all-inclusive. I'm going to introduce you to each of those groups, and then we're going to learn about a few of those organisms. So to get us started talking about these eukaryotes, early taxonomy schemes grouped organisms based on structural similarities. Remember, we talked about taxonomy before, and the modern classification is based more on similarities in nucleotide sequences. So once we learned more how to do genetic sequencing and interpret that very well, we started moving some organisms around, based on their ancestry. So you'll see a few different trees on this, phylogenetic trees on this slide, and here in blue we have bacteria, then we have archaea, we've talked about those, and then eukarya. So eukarya, you know that the prokaryotic and eukaryotic cell types and the general characteristics between those two. And so we are addressing the eukarya. So those protozoa and fungi, and eventually the helminths, which are the infectious worms. And these two phylogenetic trees, I think, visualize it a little bit better for us to see where do these organisms kind of play out in the whole tree of life here. So you see down here at the bottom, we have eubacteria and archaebacteria. Then we have protista, animalia, fungi, and these are the organisms that we're going to be talking about right in here. Okay, so we're jumping into the group known as protozoa. So first of all, distribution of protozoa. Most will require moist environments. Most will live in ponds, streams, lakes, oceans. Others live in moist soil, beach sand, decaying organic matter. Very few protozoa are actually pathogens. We will only address the human pathogens. Some other characteristics. Most all are modal. Modal, that means that they can move around. And how they move around is one of 3 ways. Either they have cilia, or they have flagella, okay, or they can move around with what we call pseudopods. And that's more of like an amoeba moves, where you have some cytoplasmic streaming. But they are modal and able to move around. Now about the protozoa and different stages of the life cycle. What you're going to notice is when we get into the nuts and bolts and start talking about the individual species of protozoa, you're going to see some pictures of life cycles. Because with protozoa, you're going to see that there is a distinct life cycle. There's going to be some changes in the morphology of these organisms throughout their life. So one is a modal feeding stage called a trophozoite. That's how you pronounce that, trophozoite. So it's modal, means it's able to move around. And then it kind of toggles between another stage, resting stage called a cyst. So we call that a cyst. Think about it like a capsule. It's an encysted variety of it. It's a dormant resting stage when conditions are unfavorable for growth and feeding. All of the protozoa will reproduce asexually, but some will also reproduce sexually. Okay, the first one we want to discuss is Plasmodium. So Plasmodium vivax, which causes malaria. Actually, there are 5 different species of Plasmodium that can lead to malaria. I'm just giving you one example of Plasmodium vivax. It is endemic throughout the tropics and subtropics. Malaria causes more than 1 million deaths annually throughout the world. We don't often hear about malaria here in Missouri, but it is a worldwide concern. Transmission. The transmission of this particular organism is by arthropods. So arthropod-borne transmission. The specific arthropod is a mosquito, and the type of mosquito is called Anopheles. It is the Anopheles mosquito that will transmit the Plasmodium organism to humans. The incubation. Incubation is pretty wide and varied, and it is anywhere from 6 to 37 days. Signs and symptoms include fever, chills, diarrhea, headache. It can result in anemia, weakness, and fatigue, and it gradually occurs in paroxysms. This term, paroxysm, it might be a new term for you, and what a paroxysm means is that you're going to have episodes of being really, really sick, followed by episodes of feeling just fine. So you think that you're cured, you think that you're better. Lo and behold, what happens is that you get sick again. So it's kind of waxes and wanes with how sick that you actually are. That's called paroxysms. And malaria can display that. Now, I want you to take a look at the pictures that I put up on this slide. And one of the pictures, I want you to read through the 1 through 6 on the kind of the life cycle of the malarial parasite. And then I have some histio slides up there, and that will show you what it actually looks like. What does this organism look like? Some of them are showing you inside of the cells themselves, okay? And you can actually see some of the organisms over here on this bottom slide. Kind of interesting to actually see those. So take a look at that before you move on. Okay, still talking about Plasmodium that causes malaria. And we're going to get into a little bit more detail about that one. Now, the reproductive cycle hides the parasite from the immune surveillance, from our immune system constantly on surveillance, and these tend to hide from it. And what the pathogen does is it injects toxins into the host cells. Adhesions, which are types of molecules, allow red blood cells to adhere to certain tissues. And when we have red blood cells attaching to tissues, that's not good for us. Okay, so we start to feel the effects from that. So these organisms are really good at avoiding the immune cells of the immune system, and they are really good at avoiding detection, so they can hide. They hide within vesicles to avoid that detection. Now, what does this organism do? First, it travels to the liver. So first it travels to the liver and then to the red blood cells. It infects the red blood cells, and then it will burst open the red blood cells and destroy it. So you can imagine on a wide scale that we're going to see a decrease in our red blood cells. What is the function of our red blood cells? I know you know that. And to help us carry oxygen, right? It has the hemoglobin in it. It helps us to attach oxygen to carry it to our tissues. And so if we have a drastic reduction in the number of red blood cells, that can lead to anemia. It can lead to anemia, enlarged spleen, and even death. So how is this treated? It can be treated with medication, but some species can lie dormant in the liver and reactivate years later. About 300 million people, let me say that again, 300 million people a year are infected with this type of parasite. It's pretty significant. Now there is treatments. You can take anti-protozoan medication. I do have a video for you to watch, so I would like you to click on the link at the bottom of the slide and watch that video. It's a good animation to kind of bring together what I said in the slides. Our next organism we're going to talk about is Toxoplasma gondii, which causes toxoplasmosis. The transmission officially is fecal-oral, so it's fecal-oral transmission, most of the time by cat feces. It can remain viable for up to a year, right, outside of the body. Eating undercooked meat is also a way of transmission. I guess technically mice, rats, cats, and pigs. Incubation. Incubation is unknown. We don't really know how long the incubation is. What we do know is that for protozoa, they're pretty wide and varied, so this one probably is as well. Signs and symptoms. Toxoplasma gondii, when it infects humans, we will have sore throat, fever, enlarged lymph nodes, and enlarged spleen, sometimes a rash on the surface of the skin. Pathogenesis. So the pathogenesis, what we do see is that this organism will cause necrosis. So if necrosis is a new term for you, that just means cell death, tissue death. So much cell death that it causes death in the tissue, necrosis. So necrosis and tissue cysts may develop. And you remember when I first started talking about the protozoa, I said that one of the life stages in the cycle was a cyst. So what happens is that in part of that life cycle, it will encyst into certain tissues like the muscle. So a little bit more on pathogenesis. It can be deadly well, the most dangerous type of toxoplasmosis is fetal toxoplasmosis. This particular organism can cross the placenta and cause some major damage in the developing cells. So how it's dangerous, how the fetal toxoplasmosis is dangerous, it can be deadly for the fetus and result in miscarriage. Lots of times if the mother is infected within the first or second trimester of pregnancy, it can lead to that miscarriage, cell death, tissue death, and miscarriage as a result. If the mother is infected within the third trimester, usually there's a better outcome for the fetus, but the fetus sometimes will have nervous system complications, but there is a better outcome if she is infected within that third trimester. Now, this is mainly in temperate areas, and we are temperate, right? We look at the climates, we are temperate climates. So this is within our climate, it is here. Now, treatment. Medicine is available, so anti-protozoa medications, but there are some resistant strains of Toxoplasma gondii. Now, if you're not pregnant and you have been diagnosed with Toxoplasma gondii, in actuality, do they really give you the medication for this? I have been talking with others and clinicians. It doesn't seem that they really do give the medication. It's kind of self-limiting, and we let the immune system take care of it, which it usually does. Now, in a pregnant individual, that might be different. But a healthy individual, usually the immune system can kick this Toxoplasma gondii in the butt and destroy it. Okay, this slide shows a life cycle of Toxoplasma gondii, and I like that this has some nice graphics in it and helps you kind of to remember about this organism and the life cycles and the different hosts that that potentially can harbor it. So if we look at number 1 on the right side of that little kitty cat up there, it has sexual reproduction of parasite occurs in the cat's digestive system, and immature oocytes are shed in the cat feces. So that's just the cyst that we were talking about. An oocyst produces internal sporozoites. That's just the type of the type in the life cycle. All right, so if we're following along, rodent or a bird ingests the sporulated oocysts, or livestock or a human consumes them, okay, becoming an accidental host. Now, remember I said this was fecal oral, so it's coming out in the feces and somehow or other we are ingesting it. So it's either coming from the cat feces or it's coming from the cow when we actually eat the cow, saying that it wasn't cooked enough to cook the pathogen and destroy the pathogen. But what we do see is a lot of transmission actually occurs with the cat feces into the human, and human being an accidental host, it is still pathogenic to us. So what is one thing that we can do to avoid it? If the majority of the people are getting it from cat feces, it is probably, and in a healthy individual, our immune system takes care of this. It's not so bad for us usually. We just need to be very careful when we clean out litter boxes. Should we tell pregnant individuals to avoid cleaning the litter box? Probably so. It's probably a good idea to avoid cleaning litter boxes when you are pregnant, right? Just to avoid that exposure, that potential exposure. I'm not saying that every cat has Toxoplasma gondii or has that. It would be releasing it into the feces. We just don't know, especially for cats who are going out and catching mice and other types of mice and rats that could potentially have it. They can potentially have it in their feces. So we want to just tell people about it. Let them make that decision. Let them be as informed as they can possibly be. Now we're on to talk about Naegleria. So Naegleria fowleri causes primary amoebic meningoencephalitis. That is a mouthful. But Naegleria fowleri is the organism. Now this is a protozoa and it's in a type of an amoeba, right? So we have amoebic meningoencephalitis. Now transmission is waterborne, and it is transmitted to humans by the humans swimming in warm, fresh water. Now, this organism, Naegleria fowleri, is normally found, let's say, on the banks of freshwater systems in the soil. So this is an amoeba, usually found in a warm, moist soil on the banks of these waterways. And by washing into the waterway, that's how it gets into the water system. So about this organism must penetrate the nasal mucous membranes of the human host in order to cause harm, in order to infect the human. What we do see is that when the temperature, about the air temperature or the water temperature gets to be about 82 degrees, at least 82 degrees, these organisms become we find quite a bit of them. So they start to multiply a whole lot within the water systems. So unless that water is 82 degrees or higher, that's when we start to see the problems from this organism, because they are so prevalent if they happen to be there. So what happens is that someone goes swimming. All right, so someone goes swimming in a waterway, freshwater, right? In the freshwater, and they're diving in, or they're doing some flips, and that water, which happened to have the Naegleria fowleri in it, goes up their nose. You've probably been swimming before and this happened to you. You jump in the water, it's going to go up your nose. You do some flips in the water, it's going to go up your nose. You're splashing and it's depositing it up into the very, very roof up here to the epithelium, all right, to that olfactory epithelium, if you guys remember that from AMP. And when that water happens to have Naegleria fowleri in it, that amoeba, if we deposit it into that area, that's the first step in infecting ourselves, unfortunately. So incubation is 1 to 2 days. Let me say that again, 1 to 2 days, and it's definitely less than 1 week. Okay. Signs and symptoms. Signs and symptoms are a headache, a severe headache, fever, a stiff neck, vomiting, confusion, and seizures. This happens right away, almost usually within the first day or two of infection. So why is this so dangerous? Like I said, we have to deposit that organism to that olfactory epithelium. If you remember the cells that are at the olfactory epithelium, this is where the olfactory nerves, this is where the nerves that lead to the olfactory nerves are. And so it's going to penetrate into the skull along the olfactory nerve. It's going to travel along the olfactory nerve to the brain. And then there at the brain, it's going to start destroying nerve tissue. So this amoeba starts destroying nerve tissue. And as you know, once the nerve tissue, neurons in the brain start to be destroyed, we don't replace them that quickly. We don't replace them at all, if at all. And so it starts destroying brain tissue almost immediately when it gets there. Hence the seizures, hence the headaches, right? It's infecting the brain. Okay, treatment. So treatment is largely unsuccessful unless it's caught quickly. This has to be caught quickly. They start experiencing these signs and symptoms within a day or two. And historically, those signs and symptoms have probably been attributed to something else. Okay, something else, some other type of pathophysiology. And this is rare. This is actually a very rare disease, rare infection in humans. But the more that we know that it happens, the more educated that our clinicians are getting about these more rare diseases, we can get some better diagnoses for individuals. So yes, it is rare. Naegleria fowleri infections are rare. In the 10-year span from 2009 to 2018, 34 infections were reported in the United States. So, you know, overall, that's not a whole lot, but if you know what to look for, if the clinicians that they absolutely know what to look for, and get a very quick diagnosis for these individuals, then they can save them. This didn't used to be the case. A lot of people would die and we wouldn't find out until later on that they were infected with this because of an autopsy. So, how were they? Just looking at that surveillance. 30 people were infected. Of those cases, 30 people were infected by recreational water, right? Jumping in the water, happened to have the amoeba, got deposited in the epithelium, in the olfactory epithelium. 3 people were infected after performing nasal irrigation using contaminated tap water. And 1 person was infected by contaminated tap water used in a backyard slip and slide. So if you've been on the slip and slide, you know that water gets deposited right up in the nasal passage. Now back to this nasal irrigation. This nasal irrigation, I want to tell you a little story that goes along with that one. So after Katrina hit, and this is an example of one of those individuals. After Katrina hit in the New Orleans area, of course it was devastated, and lots of the plumbing, the water pipes were devastated as well. Once they got everything up and running again, there was a little boy who died, and as it turned out, from autopsy, they discovered that he was infected with the Naegleria fowleri. This is also known as the brain-eating amoeba, if you've heard of that. So they tried to figure out he hadn't gone swimming. He wasn't anywhere. There was no, how we normally think of it, we think of somebody who goes swimming, who's diving in the water, who's playing in the water, and the water gets deposited, right? And because of the epidemiological study, they figured out that he was getting nasal rinses. So if any of you guys have ever had that done, so the neti pot, right? They have these neti pots, you put some saline in it, or they have the little squeeze bottle for people who have allergies, maybe a sinus infection to help irrigate out your sinuses. And that's what he was doing. His mother was helping him do this because he was having some sinus issues. And it turned out that was the only thing that he was doing. And so they were checking the water. They checked the water, and sure enough, they found Naegleria fowleri. They traced it back, and they found a break in one of the water lines, and the amoeba was coming from the soil. It came from the soil, entered into the water lines, and lo and behold, with the nasal irrigation, it deposited to the olfactory epithelium, and the little boy died, unfortunately. So that does happen. Now, when this came out, I remember vividly, listening to that story, reading about it, and going, I have one of those irrigations. So I went down and I read the box really quick, and I'm like, what? This can't happen. How can this happen? Because it's saline. Saline, you don't think of that saline as being a way. Anyway, so I read it, and sure enough, it says you need to boil that water. So you're going to boil the water before you use it in the nasal irrigation, just in case there's any pathogens happen to be in there. So now I have two links for you. So at the bottom of this slide, I would like you to check out the first link. The first link is kind of a collection area from one of the parents of someone who has died before, of a child who has died. They tell the stories of these individuals that have, accidentally been infected with Naegleria fowleri, and their outcomes and their stories. I would like you to check that out. And then the second one is a link a few years ago, and this was an area in South Carolina, and there was an outbreak in South Carolina. So I think this was in 2016. I had gotten an email from a former student, and she wanted to tell me about what was going on in her hometown. And what had happened, there was an outbreak, and someone got infected with Naegleria fowleri, and it was, of course, devastating to their area. And at the time, this is very rare, right? These organisms are pretty rare. And, she had taken my microbiology class, and we had talked about this in class. And she wanted to tell me that when this all came out, at this outbreak, that no one knew anything about this organism, had ever heard of the organism before, but that we had covered it in class, and that she wanted to thank me. We covered it in class because now she was kind of her resident expert in this particular organism. So she was able to help educate others on the transmission and that the infection and things like that. So kind of thinking forward for you guys are now ambassadors of everything microbiology, right? So you just kind of keep that in mind, and going forward to educate and inform others of things like this. All right, so now we want to learn about another type of amoeba, and that is Entamoeba histolytica, and that causes amoebiasis, and it's more generally known as amoebic dysentery. So the transmission of Entamoeba histolytica is fecal-oral. So fecal-oral transmission, and how do humans usually get this ingested, is by fecally contaminated food or water. Now, incubation is wide and varied, just like for other protozoa. It can be anywhere from 2 days to months. Signs and symptoms, and here's where we get into many are asymptomatic. So a lot of people who are infected with this particular amoeba are asymptomatic. That means they are lacking of signs and symptoms. Those who are symptomatic will have chronic mild diarrhea, cramps, colitis, and necrotic lesions in their intestines. So diarrhea, cramps, colitis, that can be attributed to lots of different things. Sometimes it can be Entamoeba histolytica. So pathogenesis, what does this organism do? or what does it do to the body? It will actually produce some proteins that are toxic to cells. And that's what facilitates the invasion into the intestinal cells. Now, I have necrotic lesions. Necrotic, think necrosis, right? Tissue death. It injures the cells, it injures the tissues. And what I have here on this picture, right here, this picture, is a large intestine that has been prosected, folded open so that you can see it. Now, a large intestine usually doesn't look that way, and I want you to look at the dark places and the holes in it. Those are the necrotic lesions. That means it's caused cell death and the tissue death. And when something causes a hole in the large intestine, that's pretty severe. And what can happen is feces will leak into that area and cause lots and lots of infections and even death. So Entamoeba histolytica can cause long-term diarrhea, right? This chronic diarrhea and cramps and colitis that a lot of times is attributed to something else. And then it can actually be pretty severe, right? When it can actually cause holes or necrosis in our large intestines and then therefore make us septic. Okay, treatment. There is anti-protozoa medication that can treat this type of amoeba. The key is the correct diagnosis. Okay, our next organism we're going to learn about is Trichomonas vaginalis, and it causes trichomoniasis, or a slang term of trick. So there is a video that I would like you to watch. I want you to click on that link, and I want you to watch that short video on studying trick. Then come back to this slide. So hopefully you were able to watch that video. It gives a good introduction to the Trichomonas vaginalis. And I want to give you a little bit more information about this organism. It's a pear-shaped organism. You can kind of look at the picture to my side. A pear-shaped organism, and it moves via flagella. So you can see that flagella in the picture, I believe. Now, transmission, how is it transmitted? It's transmitted via sexual intercourse. So this is a sexually transmitted infection. Incubation, this is a protozoa, so you probably think it's wide and varied, and it is. Incubation is going to be anywhere from 4 to 20 days. Signs and symptoms. So signs and symptoms of this type of infection for females. For females, you're going to have a very itchy vulva, burning vagina, frothy, smelly, yellow to green discharge, and lots of redness. Now for males, typically they are asymptomatic. Males tend to be asymptomatic, so they lack the typical signs and symptoms. When males do have symptoms, they experience penile discharge, painful urination, and pain in the testes. So this particular organism is quite severe, right? So when it does have symptoms, it is quite severe. And it's pretty wild to think that some people will be infected with this organism and have no signs or symptoms whatsoever. Okay, on to the next slide. We're going to learn even more about Trichomonas vaginalis. Okay, a little more about Trichomonas vaginalis. We're going to talk about the pathogenesis a little bit and what this organism does. So this organism will adhere to the epithelium. And inside the vagina or inside the urethra. And it's the vagina for females and usually the urethra for the males. And it's going to cause tissue damage because of that adhesion to those cells. Now the froth that is produced. I said that you could have some yellow to green frothy discharge. And that froth is the gas that's produced by the organisms. What does this organism do? It adheres, the cells will eventually die, and it's going to consume those cells. So that's what this organism munches on. It consumes dead cells. Now, our white blood cells. Our white blood cells are alerted that there is an infection, something going on, there's an invader, and so you're going to have lots of inflammation. You're going to have lots of inflammation that's going to account for the swelling and the redness and the itchiness, and you're going to sometimes experience fever. Now, what can happen? Long-term inflammation. So a lot of times people are infected with this organism and for a very long period of time, and because they don't even know it, right? They're infected with it and they don't know it, or they attribute it to something else, certainly not Trichomonas vaginalis. And so if you have this long-term inflammation, that can lead to some pretty severe consequences. It can lead to preterm labor in females, cervical cancer in females. In men, long-term inflammation in that area can lead to prostate cancer. So pretty devastating. Now, treatments. We have some anti-protozoa medication. A few of the Trichomonas vaginalis strains are resistant to the medication. And so it's a little bit harder to treat. Now, there's another video that I would like you to watch. It's titled "What is Trichomoniasis?" It's on the Khan Academy and it's on the YouTube. So I want you to just follow the link on your slide. And I like this one because it reinforces what I talk about here, and you get to see some pictures. And it, like I said, it reinforces the information that we covered for Trichomonas vaginalis. So please watch that video, study that video, and then move on to our next slide. All right, the next organism we're going to learn about is Cryptosporidium parvum, which causes cryptosporidiosis. So transmission of Cryptosporidium parvum is waterborne. And it's basically because of fecally contaminated water and sometimes fecal contaminated food. This can be contaminated surfaces as well. Now, another interesting tidbit about this particular organism, it can survive 6 months in the water or the food. That's pretty long comparatively. It is a zoonotic disease, so it can be spread from different animals. And it likely is spread from different animals and mainly the mammals. Okay, zoonotic, but through mammals. Incubation. Incubation is about 6 days for Cryptosporidium parvum. And signs and symptoms. So signs and symptoms include fever, loss of appetite, nausea, abdominal pain, diarrhea, headache. And what you'll notice, there's a few things on this slide I want you to look at. First is the histio slide, and it's showing what does that organism actually look like. What does that organism look like? And you can look at that circle for what does the protozoa look like. The second one here, right next to me, is the outbreaks from 2009 to 2017. This is our best estimation on the latest data, right, from the CDC. So outbreaks of diarrhea most commonly linked to pools, cattle, childcare, so daycare centers. So you'll notice the percentages there. So pools about 35%. How does it happen in pools? Well, this means that someone's infected with the organism and they are experiencing this diarrhea. So maybe some of it's coming out and into the water, and then you get infected with it. So cattle, 15%, not too much, but remember I said that sometimes with food. So this is that if you're touching the contaminated meat. So let's say that this is a zoonotic disease. So the cattle is acting as the host and it's infected with it. It gets slaughtered, it's in the meat. And if we don't cook that meat long enough in order to destroy the pathogen, then we can be infected with it ourselves. Childcare, 13%. So this means that a child is sick with Cryptosporidium parvum and goes to the daycare. And children and little babies are really, when you have a lot of diarrhea and they're sick, their hands tend to go into the diaper, and then now it's on their hands and it gets spread around very easily. So childcare, about 13% of those. The next slide describes a little bit more about Cryptosporidium parvum. Now a little bit more on Cryptosporidium parvum. This particular organism infects the epithelial cells of the small intestine. And when that happens, let's think about this. It infects the epithelial cells of the small intestine, and we think about what is the major function of those cells, and one of them is absorption, right? Absorption and secretion. So if it's going to infect those cells will not be able to perform their normal function of absorption. So absorption in that human host decreases, and that can lead to what we call malabsorption. And also secretion. So if that cell is, one of its jobs is to secrete certain substances for digestion, it's not going to work as well. Now, because it is destroying those cells, it infects them and destroys them, secretion of water and electrolytes increases into the small intestine. And so what does this result in? It can result in pancreatitis, and it can result in what we talked about, those normal signs and symptoms of diarrhea. Now, another bit of information about this organism. The infectious dose is very low. So what does it mean by infectious dose? This means how many or how much of this pathogen needs to get inside of our body in order to result in an infection. So we call that an infectious dose. For this particular protozoa, it's very low, and we only need about 10 oocysts at stage of life cycle in order to infect humans. Only 10. That's not very much. That's not very much. Another tidbit of information about Cryptosporidium parvum is that it is resistant to chlorine. That's not very good for us. We think about the typical transmission. We know this is fecal oral, but it's usually spread in waterways. So waterborne. There is medication to treat it. But we need to catch it very quickly before malabsorption and dehydration occurs in that individual, and which can actually lead to death. Now, there in 1993 in Milwaukee, there was an outbreak of Cryptosporidium parvum, and it actually got into the city's water system. Okay, and it caused this huge outbreak. And so Michigan gets their water from, Milwaukee gets their water from Lake Michigan. And it was thought that, it was from the runoff of infected cows. So if you have some infected cows and you have lots of water from rainfall rushing over those fields and leading into Lake Michigan, that contaminates the water there. And then Milwaukee was pulling that water for their water treatment plants. And notice I said that it was, a lot of these strains are resistant to chlorine. So even if they were chlorinating that water before they sent it out to the residents, it may or may not have killed this particular organism. So it takes the cyst in that life cycle in order to infect the human, right? And what happens is, since there's such a low infective dose, that it doesn't take much to infect a whole city. So in 1993, 403,000 people in Milwaukee were infected with Cryptosporidium parvum, and about 100 people died because of that outbreak. So it can be pretty severe. The next organism we're going to learn about is Giardia intestinalis, which causes giardiasis. Now, an older name for this was Giardia lamblia, but they have since changed it to intestinalis. So Giardia intestinalis. Now, transmission. The transmission of this protozoa is waterborne. So it's fecal oral, but fecally contaminated water is the most common type of transmission. Incubation is 6 to 20 days. And signs and symptoms of this type of infection in the human is indigestion, gas, nausea, vomiting, diarrhea, cramps, and weight loss. So if you have indigestion, gas, nausea, you probably don't want to eat a whole lot, and so that results in some weight loss. Now, pathogenesis. Pathogenesis is what's included in that is these organisms will attach to the epithelial cells of the small intestine. They have an adhesive disc. And if you take a look at the very top picture, okay, there's two individuals and it almost looks cartoonish. So Giardia really almost looks cartoonish, like somebody made that up, but that's actually what they look like. And you can see the darker purple on that picture, and that's an adhesive disc. That's an adhesive disc that they use to suction. It's kind of like suction cups that they use to hold on to the cells that are in the small intestine. So they have the flagella. You can see that as well. They're able to move around within the mucus of the small intestine, but it can also adhere to those cells using that adhesive disc. These organisms can travel to the gallbladder. What they do is they block absorption and secretion of certain products. We just talked about how that can affect the small intestine. Giardia intestinalis has a wide host range. And so I said it's fecal oral, but it's mainly transmitted in waterborne. It's mainly waterborne, so fecally contaminated water. Another name for this, kind of an old-fashioned one, they used to call it beaver fever. So giardiasis, they used to call that beaver fever. So if you were swimming in an area, if you were in a lake or a stream that had beavers, then a lot of times the beavers would be infected with it. And so they would attribute that to the giardia. So swimming with the beavers, lo and behold, you get a fever, beaver fever. That's how that relates. It was just that that particular organism was infected with Giardia intestinalis and transmitted it to the human. But many mammals can be infected with Giardia intestinalis, and therefore we think wild animals, and therefore the feces leads to the lake or stream. If we're in there and we ingest it, that can lead to infection. Since this is fecal oral, of course it can be on your hands from the feces and get administered that way anytime you touch your face or ingest it that way in any portal of entry as well. Okay, another characteristic about this organism is that it is resistant to chlorine. So it is resistant to chlorine. So we think about, well, we would like to think we're pretty safe in swimming pools because it does have chlorine in drinking water. However, if it is resistant to chlorine, a lot of times it won't kill it. So we have to kind of think about that as well. There is medication for it, antiprotozoan medication. The key is accurate diagnosis, however, of Giardia intestinalis. So if we have indigestion, we have some gas, nausea, vomiting, how often is it really attributed to a protozoan? Probably not too often. So accurate diagnosis is the key there. All right, so that's it of the protozoa that we are going to discuss and talk about. And like I said, it's not an all-inclusive list of pathogenic protozoa. Just have to kind of pick and choose which ones we're going to cover. We don't have time to cover all of them. And same thing goes for our fungal infections. So the microbes that can lead to fungal infections in the human host. We're just going to discuss a few of these, but there are many, many more that are pathogenic. I wish we had time. I wish we had time to cover all of them, but unfortunately we do not. All right, so we're going to start off talking about some fungal, some certain types of fungi that can lead to infections in humans. And the first one we're going to talk about is one that you've heard about before. And we talked about it when we were talking about antibiotics. But anyway, well, let's get started. So the first one is Candida albicans. So Candida albicans causes vulvovaginal candidiasis, another mouthful. It can also cause what we call candidal skin infections, and in the mouth called thrush. So transmission. Now this one's different because Candida albicans is a normal part of your normal microbiota. So we've talked about this one before. And really, it's what causes the infection per se is an overgrowth of this particular organism, of this fungi, and because of the suppression of other types of organisms like bacteria. So when we suppress bacteria, we give a lot of room for growth for the Candida, and the most typical species is Candida albicans. And so it's an overgrowth of that population. Okay, incubation. Well, it's not so much of an incubation, but while we're taking the antibiotics, while we're suppressing other groups like bacteria, that will allow it for, I guess, our typical incubation, what we think of incubation. Signs and symptoms. So signs and symptoms of this type of infection include lots of redness, swelling, itching, and we had talked about how that is as a result of the overgrowth. And that is as a result of our immune system reacting to the overgrowth. So the redness, the erythema, I'm saying that wrong, erythema, the swelling, the itching, that's all coming from our immune system, and it is causing that to happen in our skin. Okay, we can also have the thick white or gray discharge. Now the thick white or gray, now that is the fungi. Okay, that part is the fungi, but the bright redness and the itchiness and the swelling, that's all as a result of the immune system or the natural resistance there trying to take down that population of Candida. It's what we call the inflammatory response. Now treatment, there is medication that treats it. They're called antifungals. Now there is, and I have a couple of pictures up here. So this looks a little way different. This is a fungal infection. So the very, very top picture up there is a slide of what it looks like. And of course here we have a little baby bottom we have some overgrowth. You see the bright redness. Now we're showing some thrush, right? So we actually see the growth of that fungi there. Now there's another one that you may have heard in the news, still the same genus, Candida, but it's an up-and-coming organism that's causing infections, and it's called Candida auris. Candida auris, which is an up-and-comer as our healthcare-associated infection. So we're seeing a lot of the spread of this particular organism in healthcare facilities. What's different about Candida auris is that it is more pathogenic than our Candida albicans. We know a lot about our Candida albicans, and usually we can treat it with medications, not really a problem. Other than that, no, I should say it is a problem, but not really life-threatening. Now, Candida auris is something different, and it is more virulent. It has more virulent factors, and it can actually enter into the bloodstream. So if it's transmitted in healthcare establishments and it causes an infection in these patients and it's getting into the bloodstream and it's actually killing some of these individuals. So that's one that you might start hearing a little bit more from. It's called Candida auris. Okay, so on to some more fungi. Now, fungi in the genre, that means the genus, Epidermophyton, Microsporum, and Trichophyton. Those three cause ringworm, athlete's foot, and jock itch, respectively. You may have heard when referring to this type of infection or organism that leads to that infection is tinea. So tinea is the name of a group of diseases caused from this type of fungi. Collectively, they are also called dermatophytes. Dermatophytes are not a scientific name. The dermatophytes, that's just a kind of a, I don't know a better term for that, kind of a marketing term, dermatophytes. It's a way to talk about the tinea, and the tinea are the genre that are included in there. So a little bit about these. They will grow on the skin. So they will grow on the skin, the nails, and the hair. A little less on the hair, and mainly on the skin and nails. Transmission. Transmission for these types of fungi is direct contact, and it may be present in soils and or on pets. Okay, so a lot of times we will get it from the soil, or your pets are outside exposed to the soil and your pets get it, and you can get it from your pets. So warm areas of the body, warm, wet areas of the body are more prone to the infection by fungi, and that is just in fungi in general. Now, incubation. Incubation varies for fungi. We can't narrow it down like we do some other types of organisms. Signs and symptoms. A lot has to do with the inflammatory response. So itching, redness, erythema. For ringworm specifically, it produces irregular rings on the surface of the skin. So you can see the top two pictures that I put up there. This poor individual has lots of ringworm all over their torso, and kind of zoomed in, you can see what that actually looks like. So that ringworm. I tell the story in class sometimes. I think I was about 16 years old, and on my leg, I got a spot that kind of looked like this. And my mom was looking at it, and she's like, "Oh, I think it's ringworm." And of course, my 16-year-old self did not know anything about, ringworm. But when you say worm, I'm thinking that it's a ringed worm, right, in the surface in your skin. So I freaked out a little bit, and I think I may have cried and was very upset about that until they explained that no, it's not a worm, it's not in my skin. This is just it's a fungal infection. So not quite as upset, but still a fungal infection. Now, so pathogenesis, what's going on here? This type of fungi will invade the dermis. Remember the layers of the integumentary system? So we'll invade the dermis and the hypodermis. There's other names for the hypodermis. You may know it as the subcutaneous tissue. So it will invade the dermis and the hypodermis. Those are for the deeper layers of our skin. It produces an enzyme known as keratinase. So notice the ASE at the end of that, ASE. A lot of times that's going to be your tip off that is an enzyme that breaks down keratin. So there are two types of keratin in our body. There is hard and soft keratin. Hard keratin is found in our hair. The soft keratin is found in our skin. So the fungus will actually eat keratin. It eats the keratin that our keratinocytes make, and which is found, a lot in our dermis. So it munches on the keratin, uses the enzyme to break down the keratin, and that's how it establishes that infection. So the how do we treat this? This is treated with antifungal medication. Antifungal medication as a whole really is, we don't have too many types of antifungal medication relatively. And a lot of the antifungal, we have topical antifungal medication, and then we have some that you can take internally. Now, topical antifungal medication, because the organism is once it establishes that infection, it's within the deeper layers of the skin, which is harder to get to. However, so there is some degree of topical medication that can help us with that. Widespread infection, or kind of like the guy at the very top that has it all over, not just one little spot, gets harder to treat, and we may need internal medicine, right? We may need internal medication for that. For internal medication for fungi, what we have that will actually work against that type of infection happens to be somewhat toxic to our bodies. So we really have to be careful with it in terms of how it affects our liver. and it can cause some liver damage and in turn other types of organ damage as well. So it's not something that we can take long term, and we really have to watch how it affects our liver so it doesn't damage our liver too much to where it causes liver impairment. All right, so that is all that we are going to cover for the protozoa and the fungi. I wish we had more time to cover even more species and what they do. It's very interesting. If this topic does interest you, there's more that is covered in our textbook. And there is some parasitology. A lot of these organisms are covered in classes known as parasitology. So maybe later on in your academic career, you can take some parasitology courses if this happens to interest you. The next lecture to listen to is still on eukaryotes, but it's about helminths. And helminths is a term for what we use for parasitic worms. So on to that one.